We're delighted that Dr. Zhang chose to join our community of scholars here at Rutgers, and welcome her today as our Institute for Quantitative Biomedicine seminar speaker. Dr. Zhang's research efforts are focused on designing nucleic acid nanostructures with increasing complexity and precision, constructing dynamic DNA devices with programmable behaviors, and ultimately rebuilding synthetic biochemical molecular systems that approach the complexity and sophistication exhibited by nature itself. The title of Dr. Zhang's presentation today is Information Directed Self-Assembly of Nucleic Acid Nanostructures and Devices. Faye, it's great to have you with us. Thank you for participating under these challenging circumstances. The floor is yours.
Thank you. Thank you for the nice introduction. Hello, everyone. Thank you for coming. I'm so glad to be here to share of my research with you. Today, I'm going to talk about the information direct self-assembly of DNA and RNA, some structures, and some functional devices. Let me start my slides. So here shows a kinetin moves cargo away from a nucleus along microtubules inside cell. So the green nature creates such amazing biological systems. We have always been trying to ask big questions like, what is life? We don't know the whole answer yet, but we do know is information-rich, self-assembled, and dynamic system. So the scientific question we ask ourselves is, can we mimic nature to engineer complex nanostructures and machineries? And if so, how? So different people trying to answer this question differently. For example, in synthetic biology, people try to redesign the biological modules or systems, and they try to harness the power of nature to solve the new problems. But here, a group of people, including me, we are trying to solve this problem in a different way. Here we call ourselves biomolecular programmers because we try to create new rules to reprogram those biomolecules, to assemble them, to manipulate them in a designed way. So we are trying to create a molecular toolboxes. What is that? So imagine this. One day you will get a deliver box, a package, a box of molecular Lego you ordered online, and you can assemble it in your kitchen by a very simple one-piece instruction paper. And then you can cook it in your microwave, or you can heat it up by the warm water, and then it will assemble into the design way, which is a molecular robot to get into your body and check your health. So it will not be just a science fiction story in future, because my friends and I, the biomolecular programmers, are working on creating such molecular toolboxes to deliver to you someday. It's amazing that there are only a few building blocks that nature used to construct the complex living systems. They are nucleic acids, proteins, carbohydrates, and lipids. Among them, DNA, the genetic information carrier, actually is quite special because it has a well-known molecular structures and chemical addressabilities. The stable and specific Watson-Crick base pairing, AT-CG, occurs between complementary single-stranded DNA is a very powerful property because we can use this predictable and programmable property of DNA for nanoconstruction. I will show you immediately in the following slides. So back in 1982, Dr. Seeman, who's in NYU, the founder of DNA nanotechnology, proposed the immobile DNA junction, which is shown in the middle. So in this structure, he was able to fix the linear duplexes of DNA into two-dimensional building blocks. So the overhanging single-stranded DNA here work as sticky ends to direct the assembly of those individual tiles. For example, if you designed the sequence of this region, the single-stranded sequence, is the complementary region to the green region, then they will find each other to form the hydrogen bonds here, the hybridization, and a similarity along the other directions. You can design the similar thing, because DNA actually, the double helical DNA is a three-dimensional structure. It can control the nano construction in 3D. If you look at the detailed design of a similar to the crossed structure, this is like an advanced version than the initial proposal from Seeman, but it's still a four-arm DNA tile. In this DNA tile, it has 9 different single-strand oligos, and with the designed shape and the sequence, we were able to create the nanostructures that from those oligo sequence. So the real bench work or the wet lab work is quite straightforward. After we ordered this sequence, this DNA oligos, we just mix them in a designed buffer in a PCR tube, put them into the PCR machine, and under temperature cooling process, we can obtain the assembly of the individual tile as well as the designed 2D array within 2 hours. Here shows the final assembled structure. This is the structure be visualized under atomic force microscope. And you can see that is already like 17 years ago, we can reach such high resolution. Nowadays, the programmability of DNA has been improved dramatically. So this is a very old and classic example of two-dimensional DNA self-assembly. I will show you more in the future slides. Here is another example of DNA self-assembly. A DNA tile with a three-dimensional arrangement of sticky ends will link each other to form the higher order 3D lattice. Of course, it's a DNA designer crystals, which means before we grow the crystal, we know what kind of 3D lattice we will get. All the unit cell parameters and all the symmetry parameters, we got that predicted parameter before we do the experiments. If we got something wrong, or if we grow different type of lattice, it's a failed experiment because it's not the designed way. So in this case, we were able to create the 2D lattice with hexagonal prism. And you will see this is the optical image of this native crystal on the left and the bromine derivative crystal. And the morphology of this crystal is hexagonal prism, and the density map of the crystal also shows a great match with our design model. Okay, let's move on. For this slide is unpublished data. It's still ongoing progress because such designer DNA crystal, we can use it as 3D scaffold as a host to introduce gas molecule into the lattice. Since every nucleotide in that 3D lattice is under our control, we can design certain sequence. For example, here, we insert this AATT sequence into the lattice, which is a place can bind a minor groove binder on here. And if we insert that specific sequence into our three-dimensional lattice, we were able to host this dye binder into our lattice. And the density of that binder also can be reviewed in our density very well. So we hope that this is just a very initial demonstration. We hope that in future we can create a variety of DNA 3D lattices with more controllable cavities and a lot of different symmetries and a larger space to host a larger guest molecules, even like proteins, especially for those proteins that cannot be crystallized by themselves directly or easily. That actually, this idea is a very classic idea. Is the initial proposal from Seeman more than 40 years ago. But it turns out it's a very challenging task. It's almost one of the most challenging tasks in DNA nanotechnology. Because as you can see, our field actually has been grown dramatically in past 10 to 15 years due to the new technology, actually due to the new technique, which called DNA origami. I have to emphasize that DNA origami is just one branch of DNA nanotechnology because we have so many other techniques like designer DNA crystal, 2D lattice, DNA tiles. But DNA origami seems the one like really famous. So this technique is invented by Paul Rothman from Caltech in 2006. I will show you why DNA origami is so successful. This technique involves a long scaffolded DNA. Showed it here. It can be like a genomic DNA. It can be any sequence actually, as long as it's a long, we call it a scaffold DNA. And then we will add like hundreds of short helper DNA. It's synthetic short DNA with designed sequence around, like with length around 15 to 60 nucleotides long. And those short synthetic DNA will be designed to be complementary to some of the region of that larger scaffold, which means here's the one example, this red stable strand, half of that red stable strand will bind to here, and another half will bind to here. So this leads to the folding, we call the folding of that scaffold. The short red strand will bring some of the part of the scaffold close, right? And imagine if you have many of these short oligos, they will work together to bring this long.
Bear with us, folks. Our speaker seems to have locked up.
And the short DNA oligos, like the glue or the staples to stick this DNA paper together to a designed shape. So this technique is so successful because the formation yield is so high and it's so straightforward. And since 2006, the two-dimensional DNA origami technique was invented. So many other origami was created as well. For example, the 3D DNA origami was published 2009, and the curvature was published in the same year. And also they pushed the complexity of that technique to 2011 by demonstrating a complicated shape like this. So by that time I was thinking, can we do more? Can we always do more? You know, the DNA, the programmability of DNA seems such a powerful property that we can use to manipulate nucleotides or manipulate things at nanoscale. So the question I ask myself is, can we use DNA to form freely, like any shape in 2D? Like something I put it here, like 2D drawing, right? With all the freedoms. Or like something in 3D printing, you know, any shape you put it there, as long as you provide the blueprint or the map, it will generate the shape at nanoscales. So here's a strategy I proposed here. I think if we look at the previous published DNA Origami structure, you will find that one of the major limitations is all those scaffold, all these helix bundles has to be arranged in a parallel way, right? In a parallel way, here's them here, here, and then here. So can we treat any nanoscale shape with like a mesh, which means a graph consists of vertices and edges. It can be 2D and it can be 3D. I have to create a network and then use DNA to fold into the targeted network. So there are two major challenges by exploring this technique. So first of all is how to translate any like this network into a scaffold routing pathway. So we can solve this problem by applying graph theory directly to double the edge to create those unique positions. And the second challenge is how to translate this, this graph to a real three-dimensional DNA helical structures with the scaffold routing as well as the stapler strands. So I will not go to the detail of each of that, but I will show you just 3 examples. For example, if we want to create this honeycomb structure, which I laid a scaffold routing here, you will meet different situations. For example, in number 1, you will have certain edge with 2 double helical structures, and the second 2 with a crossover or bridge, we call it, in the center, and then the third is a certain terminal structures. And we lay out the scaffold routing as well as a staple arrangement in those 3 cases, and also translate it into the real B-form double helical of DNA. One more thing I want to point it out that here I didn't show, but in here you will see DNA is a vector, which means it has a 5' as well as a 3'. In the routing and the translation, you also should think about the vector aspect of DNA. So those are just the main key technique points. And after we solved all these challenges, actually, we were able to create the designed shape. For here, we are using a scaffold DNA called M13. Is a genomic DNA of a virus, and then it has the scaffold routing way showed here. And at the bottom, you cannot see the details, but all the colors, different colors, show the different single-strand DNA with the 3D helical conformations. And we use atomic force microscope to demonstrate the successful formation of our structures. The scale bar here is 100 nanometer, and zoom-in structure shows the details of that pattern, and the zoom-out structure shows the formation yield. We demonstrate the yield by AFM as well as by gels. We use gels to show the major band formation, both both methods gives us like around 80% of good formation of individual structures. And as we proposed before, we want to demonstrate that this new design method has the power that we can break the local periodicity rather than using the parallel helical arrangement. We can arrange the scaffold routing in any designed way. So we start from the honeycomb array or honeycomb shape to these three shapes. We call they are like a quasi-crystalline shapes. It's just some represented structures. And for example, this central one is Penrose tiling, right? So just one piece of the Penrose tiling. It's the same Penrose. The Roger Penrose is who won the Nobel Prize this year, but he won the Nobel Prize in physics for discovering the black hole formation. But this pattern is also named after him. We call the center of Penrose tiling, which can fulfill two-dimensional array without any gap with five-fold symmetry. Okay. And we also introduced a curvature into our design. For example, this vivid like a grid, or like a network looks like this, or even a small circle here. So after creating all those patterns, I think we are able to test our design method to producing patterns of extreme complexity, like we designed something like this. So this is a random like drawing I searched on the internet. And so by that time, I just designed this in a software called TMAT, which helps you to draw the three-dimensional helical of DNA easily, but it cannot do the automate design. You still draw it literally one by one. I draw this shape in DNA from left to right. So after I finished the flower part, I found I was running out of DNA. So I have to change the bird to a hummingbird. If you look closer, you will see each of these vertices will be a multi-arm junction. And this black strand are the scaffold strand, and those colorful strands are the staple strand. So in this whole structure, we involved 41 different vertices and arm lengths ranging from 7 bases to 121 bases. And here shows all the individual vertices, different conformation, different angles. It's a really, really detailed design because I want to replicate that that figure, like whether we can demonstrate our design strategy. Do we have that controllability or programmability? So the final AFM images shows a very satisfying result, because if you compare the design model on left to the final form structure, they matched really well. They revealed a lot of details of the design. One interesting I want to point out, if you look at this flower, actually it's a mirrored shape than the design model, because I found that at this position, at this position is a weak point for this structure. When I image those structures, I have to deposit on mica. So the whole flower part can be flipped facing up or facing down, because that is a very flexible part. If you look at different things, you will see this flower has different conformations in each of these images. So after the successful demonstration of the formation of 2D shapes, we are thinking about whether we can form 3D shapes. Here we used two strategies to generate 3D architectures. The first strategy we call the open and fold strategy. Basically, we cut through those edges to translate a 3D polyhedron to a 2D open networks. And the second method we used is the Schlegel diagram, which is the topological equivalent 2D net of a 3D polyhedron. And imagine you just have the projection of all the 2D networks, you flatten them on 2D. So it's the equivalent 2D networks. So we were able to route our scaffold by using the previous two methods to translate our 2D design method to 3D. And we were also successful be able to demonstrate the successful formation of this by using AFM images as well as cryo images. By that time, the resolution is not that good. We were just able to see some of the symmetry of that snowball cube is the name of this 3D polyhedrons. But this is actually 5 years ago. We have several following work I will mention briefly in the next few slides to show you we really get the 3D shape of what we designed. So actually the real power, or one of the most important property of DNA, is we can create a dynamic structure from it. One of the strategy we have been using for a lot is we call the strand displacement, which means if you have a half hybridized pair, for example, this orange strand complementary with the green strand partially, but you left some unpaired region here, we call toehold. And then a longer single strand DNA, which is fully complementary to the orange strand, is competitor, will bind to the toehold region. And this junction part, they will migrate along the orange strand until to the point that the purple one will fully complementary to the orange one, which will release the shorter strand. And since they are fully complementary between this purple and orange strand, the green strand will never have a chance to go back to replace the purple one. So it looks like a chemical reaction, right? If you treat this purple one as A, this as B, as a C plus a CD, and then you got to kick out of this brown one and form a new compound here. So it's a very simple and how to say powerful property, because a lot of actually a new branch they call the DNA computing was established on this slides. We are not focus on that, but today I will show you to use the DNA strand displacement reaction to do the reconfiguration of DNA structures. As I mentioned before, for the 3D polyhedrons can be constructed by the open and the fold strategy, which means for the 3D connection of this polyhedron, those two edges are supposed to be complementary because we just route the scaffold like the 2D net way. But in reality, if we want to form the 3-dimensional shape, we should close those two edges by adding the staple strand, which is like on the top. You see all these connections like the glue to glue the edges together to form the 3Ds. But imagine if you use a longer complementary strand, which will be able to replace those glues, those bridging DNA strand. It will be able to open those edges. And imagine that if you can you can also use strand displacement to get rid of those blocker strand, and then you can add a new glue strand to put it back. Basically, DNA strand displacement reaction will allow you to manipulate the staple strand, the connection between the scaffolds, easily. So we were able to demonstrate the reconfiguration or the switch between those two structures, the 3D conformation or the opened networks. Okay, that is the end of the story. In that paper that was published 5 years ago, we demonstrated that scaffold routing, and I mentioned for the flower and the bird shape, I drew manually by my hand. So even after the publication, we received so many requests because that technique is so complicated. Like few designers, like a real be the fundamental designers in our field, was able to draw or able to use that techniques. And it's not good. We don't want a technique just limited by few people. So we collaborate with computer scientists from MIT. We created this automated design pipeline. We also published the software from it. So from here, we hide all the scaffold routing and the design details inside of the software. For the user, you just need a target polyhedron. So we are targeting polyhedron because the software provides that ability by that time. You need a polyhedron as your target, and then the software will do all the design in the computing behind you, and just to give you the sequence that you need. You can use the sequence to order the synthetic DNA, then you just mix them together and cook it in your lab, you will get what you want. Here is the name of the software, is open sources. You can Google it, it will link you to the software. It's totally free. And it allows you to design the polyhedrons and the different 3D shapes. And we test our automated design pipeline and software by asking software to help us design some of the design shape. And with the collaboration with Wachu from Stanford University, they give us amazing like a Cryo-EM for demonstration the successful formation of the designed 3D polyhedrons, and it shows a great match with our models. And it turns out the 3D polyhedron is easier for automated design pipeline because all the scaffold routing and everything is confined on the 3D surface, right? The real power of that design method we published in 2015 is the freedom. So in last year, we finally finished this part, is the automated design in 2D. So in this software, we are able to connect the software with like any tablets, like your iPads. And then you can draw like anything, literally anything. You can even upload one of your selfie. And then it translates to the like a vertices and edge shape, a 2D network. It has to be a 2D network. And the software will recognize that 2D network and translate all the vertices and edges into the 3D helical DNA scaffold routing with the stable strand. And the final file you will have is a file with a synthetic DNA sequence. You can use those sequences to order your DNA plates, and then you just mix them together with a scaffold strand. You can get the final design shape at nanoscales. And I showed a lot of structural complexity of DNA self-assembly. Here, I will try to convince you that we are not just for fun or just for pretty shapes and nanoscale. We try to create some functional nano assemblies. For example, here shows the first here shows some early days application of DNA nanostructures. The DNA nanostructures provide a great molecular scaffolds for directed assembly of other nanoscale materials. For example, nanoparticle is a very good targets to demonstrate this. So that is in 2009, people use the DNA tubular shapes to create the nanoparticle helicals on it. And in 2012, people use a triangle DNA origami to organize 3 gold nanoparticles on it. And they also arranged gold nanorod with this triangle DNA origami with the designed angles and positions. They show the different conformation of those 2 DNA nanogold. And 2 years ago, especially recently, people are trying to create larger or long-range assembly from DNA self-assembly. Here, the assembled DNA origami linear arrays. So the DNA origami, individual DNA origami host individual gold nanorod. And then after the higher order assembly of the DNA origami, this gold nanorod will be arranged in a designed way. And also you can tune the helicals and the turns and the arrangement and the chirality of this higher order assemblies. This is like the most recent advances towards this direction. And another example of DNA self-assembly as molecular scaffolds is they use the DNA origami as cages or host to create protein cascades. And here I show 3 examples. Actually, the first author of those 2 examples, because they are both of them published like 8 years ago and 6 years ago. The first author is all in our records. He's one of my colleagues, and we graduated from the same group, and he's in Camden campus. And he joined the Rutgers 5 years ago or 6 years ago. Yeah, and he created a lot of amazing assemblies by using DNA structure as the spatial molecular scaffold to create like enzyme pathway or this multi-enzyme cascade to demonstrate the ability of nanoscale DNA structure to create the distance control and the orientation control. Is really amazing work. And another work shows we can also use a 3-dimensional DNA cage to encapsulate 2 enzyme pair inside of that cage. And this is a more recent publication, but I personally, I think it's a relatively bold stretch for DNA nanostructures, but I still think it's a good demonstration to show the potential of DNA nanostructures. For here, they want to demonstrate that DNA origami can be used as like a smart nanorobot for drug delivery. For example, they use this DNA tube as the nanorobot to deliver 4 thrombins. They encapsulate 4 thrombins inside, and they also create a lock and key structure here, which is half of this, let me see, this is cyan color, is light blue color of single strand, is the aptamer sequence of nucleolin. And the other half, the red part, is partially complementary to that aptamer sequence. So what is this forms as a lock. When nucleolins shows up, this aptamer will be released to bind to the nucleolin, and then this whole tube will be opened by the presence of nucleolin. So this shows as like a smart recognition way, shows here. And after the opening of this tube, the 4 thrombins on the surface, the inside of the DNA tubes will be exposed, will cause a thrombosis around or in the blood cell, in the around that. It is not a cell entering, it's just around the area. So like I said, it's not a real treatment, but I think it's still a good demonstration of the potential of such complicated nanoscale robots to show the recognition as well as the opening and the closing and the very accurate drug delivery, right? We are not deliver a bunch of like a thrombin. We designed 4 thrombin in one DNA origami. We will have 4 thrombins in DNA origami. We have the accuracy down to the molecules. So before the real demonstration or the real application of DNA nanotechnology, there were many challenges for that. For example, the stability of DNA, right? Normally, we need a high magnesium buffer to maintain the integrity of that origami shape. The other challenges like the toxicity and the biodistribution, immunogenicity, and many problems and challenges. So that's why I'm saying it's a bold stretch to demonstrate real drug delivery. Among them, there is a fundamental challenge, is the cost. Everything we need to calculate the cost. For DNA origami, it's a so expensive structure. Like a DNA origami, $200 per gram. For research, it's good. Like if I design an origami, I just tell my students, you design carefully, okay? So it's equals to, we try, but it's still such expensive material. The major cost of DNA origami comes from the synthetic oligos. Like I said, the long scaffold is a genomic DNA. We can obtain that easily, like not easily, like relatively with low cost. But those hundreds of stable synthetic oligos, they are the major cost of DNA origami. Each individual of DNA origami shape, they required a unique set of those synthetic oligos. And those oligos, individual of them, they have to be designed individually. And if you want to do some modification, for example, to bind or host some of your target structure, you should find the particular one or several stable strand according to that location and to change that area. So if your origami structure is large, like 7,000 nucleotide long for the scaffold strand, the set of DNA origami, like a stable strand, will also be like as large as your scaffold, because they will be fully complementary at the end, right? So so the next challenge I'm thinking, like whether I can get rid of this this stable strand. If I just use a single strand of DNA to fold any shape, the single-stranded folding will have several advantages compared to the traditional origami structure, because it will be a self-replicating structure, right? We call it a self-replicating single-strand origami, because you are using one long strand, it folds by itself. You can just shape the one of your or a few of your design in a small tube to your collaborators, and then they can do the amplification by themselves, and then they will renew and fold the target origami by themselves. And another advantage of such self-replicating DNA origami is the individual folding of single strand, they actually have a high efficiency, because DNA origami, it has a long scaffold to direct the self-assembly of those short stable strands, but still you have the missing stable problems, and sometimes you miss the formation yield. But for here, as long as you maintain your long materials, single strand DNA is good. It's the intramolecular interaction rather than the like hundreds of strands assembled together. So it will have a high yield. So in terms of disadvantage, right? This technology also has disadvantage. Before we design the single-stranded folding, one important concept we introduced here, we call crossing number. So a crossing number is a knot invariant that shows the smallest number of crossings in any diagram of the knot, representing the topological complexity of a knot. For example, here shows a knot with crossing number as 3. 1, 2, 3. So to create larger single-stranded folding structure, we should think about the topological configuration of such structure. Before our design, we looked at the natural structure, because in nature, there's many single-stranded folding of RNA rather than DNA. So what is the crossing number of single-strand RNA? Here shows a small subunit of ribosome RNA, and if you hold both ends of this complicated single-stranded folding and do the shaking like in the video, you will find that the crossing number is 0. So there is a reason behind that, because without creating any topological complexity, is easier for the folding pathway. But if we look at our DNA origami structure, here shows one of the screenshot of the design. If you look at the original of the DNA origami structure, all this cross, like I pointed here, the white and the orange show the cross here. Each of them is a crossing number, right? They shows a great topological complexity here. So can we like minimize that in complexity in the DNA origami? We tried various design strategies, and here is not our final strategy, but I think it's a good illustration for understanding our final strategy. We actually bring up like a waffle design. Look at that, it looks like a waffle. If you trace the single strand routing of this structure, the white strand goes up and down, but they maintain underneath the green part. But if you look through this direction, it still shows the B form double helical of DNA. We just create those crossovers, we call the paranamic crossovers, to change the routing of the DNAs. And it turns out actually this configuration never formed successfully because we bend and twist the DNA a lot. The final strategy we used is a compromise between the highly knotted structure with this waffle strategy. We create a paranamic axis shape like this red and blue part. And this paranamic cohesion, we call paranamic cohesion X, involves 4 single-strand DNA. And they will form attaching position and form a crosshair. So in total, we route the scaffold into 2 pieces, the red part and the blue part. And we designed the corresponding region from the red to the blue and to link those 2 pieces with minimums of this paranamic cohesions. So this work is collaborated with a DNA nanotechnology team from Harvard, and we were able to create such waffle-shaped single-stranded DNA origami with 0 crossing numbers as well. And this animation shows you the similar thing to demonstrate that we don't have any topological difficulties in our structures. And we were able to create different shapes, this diamond, heart, and triangle. And this work published 3 years ago. And after that, we think, can we create molecular knots, right? Because in the first structure, we fold DNA without any crossing numbers, without any topological difficulties. But here, we're thinking, can we create some extraordinary topological structures such as molecular knots? And with designed crossing numbers, can we manipulate DNA or allow the DNA itself to tighten itself up, which is really, really, unnatural, right? So we create such single-strand DNA and RNA knots successfully by using a very similar technique or fundamental building blocks is the paranamic cohesion. We have this gray and this blue part, and this 4th strand, I only use a cross to show that. And the very simple demonstration is a knot called 9-1. It has a crossing number as 9. The whole folding pathway looks like that. With any single-strand DNA or RNA, we designed this whole strand can be folded in half. And then first they fold this long loop stem structure like here. And the stem region are the fully complementary between those 2 halves. And those free unpaired single-strand region will be ready to find each other to form this paranamic region. So imagine you have long single-strand DNA or RNA, they fold a half, and then they will be ready to thread itself 9 times and to form the final design knot. If you trace your eye with me together, if you see this is from red to gray and white, then the other terminal is blue. This is red, blue. See, this whole strand, this is the whole double strand actually threaded itself 9 times to form the final structure. And the real helical representation of this knot was shown here. And by using this technology, actually, I hide some of the detail because I'm running out of time. We actually demonstrate several of the 2D array and lattice by using this. And we also adapt this technology to 3D. We were able to create those molecular knots in 3D. And as you can see, I'm also using the Schlegel diagram to translate this 3D polyhedra network into a 2D network. And the molecular knots is threading three-dimensionally into each of these surfaces. And for the tetrahedron showed in the first panel, it has a crossing number as 15, and this pyramid, it has crossing number as 20, and the last one, it has 25. As you can see, the crossing numbers, they are not too much. For the 2D structure, we test the ability of DNA constructions. We find that when the crossing number increases, the formation yield dropped dramatically. Yes, of course, it has to thread through, right? It has so many, it needs sequentially folding, and it has a hierarchical folding pathway. So many technical details are highlighted behind. But the major conclusion is that with the increasing of the crossing number, the formation yield dropped dramatically. And within if the crossing number is smaller than 30, we will have relatively decent formation yield, more than half, 50 successful formed designed shape. So that's why we designed the 3D structure with relatively small crossing number, and then we can obtain the cryo reconstruction for our structures. And it also shows a great match with our design. And so we treated this as the newest generation of DNA origami because we can fold any designed shape by using just one long single-strand DNA. And this new technique needs further development because it's just published 1.5 years ago, and we want, the future potentials or the future applications with this technique is still under development. We want to see the real power of this technique. Some of the things I just randomly proposed here, because the SELECT process is using the single-strand DNA, right? Now our ability actually is folding the single-stranded sequence into a design shape. And for example, if we designed a single-stranded library of DNA with some of the region with the designed shape, it can fold automatically, and some of the region a random library of DNA, and then we can select, for example, bivalent aptamer here, or we can even tune the distance, the length of those aptamers, and we can also fold single-strand structures, some region with designed shape or target objects. The other region are naturally existed three-dimensional folding. And then after we have all of them, we can observe them under Cryo-EM and other many potential usage. I don't know that yet. I'm still thinking about that, but I can see it's definitely like a new, provide several new exciting possibilities for us. Okay, a lot of fundamental design structures. I want to use my last 10 minutes or 5 minutes to rush in some of other of my previous work. Here we also tried different combination of DNA, like DNA with lipid, DNA with protein, DNA with peptides, DNA with inorganic materials. Here we showed a combination between DNA origami with silica. And there is a type of thing called diatom. It's a major group of algae found in oceans. It has a silica salvo, really amazing structures show here, like the honeycomb lattice with hierarchical shapes here, 3 layers, really amazing. We are thinking that can we transfer the controllability or the programmability of DNA, the origami, the DNA materials, translate that power to other type of more materials, right? So we try to code, basically cover the DNA origami where coding is a coding method, the DNA origami with a thin layer of silica. The method we employed here is a very classic method from the biomineralization field. It's called Stover method. They use the DNA as a template, and we employ the T-maps and TEOS as the silica source. And first they will form small clusters, will be deposited on the surface of the DNA organic surface. This is just a hypothesis, okay? So from simulation, of course, but we don't know what really happens there. But what we can observe it's really simple. We just premixed our reaction buffers and assemble DNA separately, then mix them together. They will deposit on DNA surface, and after 5 days of slow deposition, we will create a thin layer of silica, smooth thin layer of silica on the origami. The thickness of that layer only 2 nanometers. We showed the great match of the final structures. If you see if it's a naked origami and our TM is really thin because it's DNA, and it shows twisted and shrinked, due to the preparation of the sample. It dried out, right? It shrinked and on the grid. But after the silica coating, you will see under the same same TEM, it shows a much enhanced structure and maintain the rigidity of that structure while, and we even can observe the final products under SEM. There is also a lot of characterization, from the mature side to demonstrate really deposit silicon net. And I just want to briefly mention the final product we can get. It is a very rigid 3D structure. We demonstrate thickness from AFM as well as TM as well as SEM, and you can see by measuring the height of AFM, the final products with the mineral layer shows 6 nanometer height, and the original DNA, the out layer shows a 4 nanometer. It's not single layer. We designed a multiple layer there.
Faye, I'm sorry to interrupt, but you need to finish up. You're now well after 1 p.m. and we're beginning to lose the audience.
Oh, okay, yes. So it shows a great mechanical property as well. Yes, it's a good thing. So basically, that's all I want to present today. And we are looking for the future of this field, like what is next. Our goal is to use the information to direct the structure, and finally we will form the functional nanostructures and machineries. And the future challenge, I think, is the interdisciplinary research by combining the programmability from DNA with other materials. And finally, I want to thank my advisor from Dr. Hayan from Arizona State and all the collaborators. Thank you all for being here. That's all I have today. Thank you.